Hello, everyone, and welcome to today's live broadcast, CBD Treatment of Epilepsy and Autism in a Mouse Model of Dravet Syndrome, presented by Joshua Kaplan, Ph.D., a Senior Fellow in the Department of Pharmacology at the University of Washington. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want anytime you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Kaplan. I will now turn the presentation over to him.
Well, thanks for tuning in, everyone. I'm looking forward to sharing with you some of the work that I conducted in collaboration with Bill Catterall's lab and Nefi Stella's lab. We focused on looking at the medicinal benefits of cannabidiol, or CBD, in the treatment of epilepsy and autism in our mouse model of Dravet syndrome. Now, you're all aware that cannabis access is booming across the country and across the world. In the United States, there are over 10 states with legal recreational cannabis access, mostly out here on the West Coast. But there are an additional 24 states that have legal access to medicinal cannabis, as well as 16 states, mostly clustered down in the Southeast, that have cannabidiol or CBD-only laws. Now, cannabidiol is one of over 100 phytocannabinoids in the cannabis plant, and it's one of the most well-studied, the other being delta-9 THC. Now, THC is that high-inducing cannabinoid that is sought after by recreational cannabis users. And it has this high-inducing effect by acting on brain receptors known as cannabinoid type 1 and type 2 receptors, or CB1 and CB2. Now, CBD doesn't necessarily act directly on these receptors, but is thought to have a large medicinal scope, medicinal benefits by acting on a range of brain targets. And this contributes to the massive and potentially exciting medicinal benefits of cannabidiol. Now, THC also has some medicinal benefits and has been known for hundreds, if not thousands of years for having medicinal benefits in treating things like nausea, migraine, pain. Whereas cannabidiol also has a wide scope of medicinal benefits, including treating things like epilepsy, pain, anxiety, and a whole bunch of other disorders. Now, these medicinal benefits, though, are not without acute side effects necessarily. THC is known to induce things like anxiety, paranoia, whereas high doses of cannabidiol can induce drowsiness. But nonetheless, the benefits there are a large degree of benefits of using cannabis for the treatment of a variety of disorders, particularly because it has a relatively low therapeutic index. Now, the therapeutic index is essentially the difference between the therapeutic dose, so the one that's used to achieve a given medicinal benefit, compared to the toxic dose, the one that's going to have adverse side effects. So a larger therapeutic index is associated with a greater health, safety profile. Now, delta-9 THC has a particularly low therapeutic index, especially during the period when the brain is still developing. Cannabidiol, on the other hand, is thought to have a much higher therapeutic index, both during early stages of development and later into adulthood. But to be honest, this isn't all that well characterized and something that my lab will be seeking to identify and characterize moving forward. But nonetheless, Together, this suggests that cannabis, in particular cannabidiol, might be a promising therapeutic tool to treat a variety of disorders that are otherwise unresponsive to traditional medications. Now, one such disorder is a childhood epileptic disorder known as Dravet syndrome. And one famous case of an individual with Dravet syndrome is this young girl from Colorado named Charlotte. Now, Charlotte began having febrile seizures at age right around 8 months, and this characterizes the early stages of the disorder. Now, her febrile seizures increased in both frequency and complexity to the point that she was having over 300 spontaneous seizures a week. Now, her parents tried a variety of different traditional anti-epileptic medications, and if they had no effect, then they were sometimes making the seizures worse. And it wasn't until they essentially threw a Hail Mary and tried a cannabis extract from a strain known as Hippie's Disappointment, because it wasn't getting anyone high. It was rich in cannabidiol, low in THC, that she was able to get some relief for her seizures, and it reduced her seizures to practically nothing. Now, this idea that cannabidiol might be a promising anti-epileptic tool for treating seizures in otherwise treatment-resistant epilepsies has been picked up by a number of pharmaceutical companies. Now, Dravet syndrome is caused by a heterozygous mutation affecting this SCN1A gene, which encodes the voltage-gated sodium channels, the specific isoform NAV1.1, which is the dominant isoform in inhibitory neurons in the brain. So mutations affecting these channels leads to a reduction in brain inhibition, leading to things like epileptic seizures and a host of additional comorbidities. And indeed, Dravet syndrome is characterized by these epileptic seizures that, again, begin as febrile seizures and become more frequent and complex over time. But in addition to these seizures, they often have a host of additional comorbidities, including autism, which we're defining here as social interaction deficits, as well as abnormal social responses, as well as cognitive impairments and sensory integration disorders that, again, are consistent with autism. Now, recently, there has been a successful phase 3 clinical trial of cannabidiol in the form of a cannabidiol extract called Epidol-X, which was produced by GW Pharmaceuticals out of London. And they created this 98% cannabidiol extract and tested it on the spontaneous seizure rate in a patient population of Dravet syndrome. And in this study that was led by Oren Davinsky out of the New York University School of Medicine, found that cannabidiol, just shown here in these blue bars, caused a greater median reduction in the seizure frequency of Dravet syndrome patients compared to those treated with placebo. So this is really exciting because for once it suggests that there's a pharmacological tool to reduce seizures in Dravet syndrome. But nonetheless, it exposes a number of important scientific questions that we seek to answer. First, we'd like to identify if cannabidiol indeed is an effective anti-epileptic tool. Now, one of the challenges and limitations of doing clinical studies in humans is that due to ethical concerns, you cannot take these patients off of their current anti-epileptic treatments. So in these previous studies, cannabidiol was only used as an add-on therapy. So we'd first like to ask whether cannabidiol could be used as a monotherapy. On its own, is it effective at reducing seizures? And if it is, well, how is it working? What is the molecular mechanism, neural mechanism that it's having to achieve this anti-epileptic effects? And we can use techniques such as slice electrophysiology to gain mechanistic insight that's just not afforded by these human trials. And then lastly, we identify that it's an effective anti-epileptic tool. Can it also be used to treat additional aspects of the disorder? In our particular level of interest is autism. So to address these questions in the lab, we first identify the candidate gene that causes this disorder. And through genetic screening over the last few decades, it's been identified that most, if not all, of the individuals who suffer from Dravet syndrome have mutations that affect this SCN1A gene that encodes voltage-gated sodium channels. So in the lab, we create a mutant mouse that has a mutation to this SCN1A gene. We created a mouse with a heterozygous mutation affecting these voltage-gated sodium channels. And then we conduct a thorough behavioral characterization to identify whether or not these mice express similar behavioral phenotypes as that observed in human population with Dravet syndrome. So over the last decade or so, our lab and others has conducted this thorough behavioral characterization, and we've identified that these SCN1A heterozygous mice display the spontaneous seizures that characterize Dravet syndrome. Now, they're most susceptible to this spontaneous seizure rate, high spontaneous seizure rate, during this maximum period of seizure susceptibility between 21 and 28 days of life. And during this time, they're also at greater risk for sudden unexpected death in epilepsy, or SUDEP. Now, this interestingly corresponds to the time in which there's a downregulation of the voltage-gated sodium channel isoform NAV1.3 and a coincidental increase in the expression of NAV1.1 channels. However, as I've mentioned, due to this mutation affecting the SCN1A gene, there is a reduction in the expression levels of these NAV1.1 channels. This leads to a reduction in inhibition in the brain, leading to too much excitation, causing seizures and increasing risk of death. But it also increases the risk of additional behavioral comorbidities, including autism-like social deficits. Now, these studies conducted by Sung Han, who's now at the Salk Institute, identified that these SCN1A mice, heterozygous mice, show reduced sociability. And he tested this in a 3-chamber test of social interaction, where the subject mouse is placed in a chamber and given the option to interact either with a novel stranger mouse or an empty pencil cup, which we term the object. Now, wild-type mice, which you can see in these dark bars, prefer to spend their time interacting with the novel mouse compared to the empty cup. SCN1A mice, on the other hand, shown in the bluish bars, are practically indifferent. They spend an equal amount of time interacting with the stranger mouse compared to the empty cup. So this is consistent with this autism-like sociability deficit and lack of motivation to interact socially. They also have cognitive deficits, both in contextual fear learning as well as spatial memory deficits. Now, Dr. Han looked at the behavior in a contextual fear conditioning paradigm shown on the left, in which mice must learn to remember contextual cues of the environment in which they received a fearful stimulus, in this case, a single shock. And mice display their memory for this fear by freezing. So when they get nervous or scared, they freeze, and it suggests that they know that there's a fearful stimulus present. Now, wild-type mice, again shown in these dark bars, remember that they received this fearful shock in this context, and it's indicated through a high percentage of freezing, or a high rate of freezing, both 30 minutes after the shock was given and a day later. Whereas SCN1A heterozygous mice show impairments of this memory. And that's indicated by the reduction in their percent freezing at 30 minutes and 24 hours later. So they show impairments in fear learning, and they also show impairments in spatial learning, measured using the Barnes maze, in which mice learn the location of an escape box, and every time they go through another trial, quickly run to that escape box if they're able to remember where it is. Now, wild-type mice, shown in these blue circles, learn where the location of that escape box is based on cues around the room, and therefore their escape latency is reduced over a number of trials, indicative of intact learning mechanisms. Whereas SCN1A heterozygous mice have difficulty learning the location of this escape box. and struggle to decrease their escape latency over the number of trials. So together, these data suggest that our SCN1A heterozygous mice, which I'll now refer to as Dravet mice, recapitulate these core behavioral phenotypes that are seen in a human Dravet syndrome population. They have spontaneous seizures in essentially mouse adolescence, at which time they're also at greater risk for sudden unexpected death in epilepsy or SUDEP. And they also express additional comorbidities, such as autism-like sociability deficits and cognitive impairments. So importantly, this makes our Dravet mouse a powerful precision medicine tool for establishing the strong predictive validity to test cannabidiol's effects on the phenotypes that are associated with Dravet syndrome. So the next question becomes, can we achieve behavioral rescue using a pharmacological approach, in this case, cannabidiol? And we began our investigation by looking at the effect of cannabidiol on the febrile seizures that characterize the early stages of Dravet syndrome. Now, these studies were conducted by Ruth Westenbrook in the lab, and she mimicked these febrile seizures using a thermal induction protocol where she heated the core body temperature of the mouse up to 38 degrees Celsius, which is about a low-grade fever, and held them there until a seizure occurred. Now, she found that a single acute IP injection of cannabidiol at 100 and 200 milligrams per kilogram reduced both the seizure duration and the severity of that seizure once it occurred. So this suggests that cannabidiol on its own is effective at protecting against the febrile seizures that characterize early stages of the disease. But we next wanted to test whether cannabidiol was effective at reducing the spontaneous seizure frequency that characterizes later stages of Dravet syndrome. So to do this, we injected mice twice daily with cannabidiol at 100 milligrams per kilogram and measured the frequency of spontaneous seizures during this period of maximum seizure susceptibility at 21 to 28 days of life. Now, vehicle-treated mice have a high rate of these spontaneous seizures, as you can see here in blue bars, whereas cannabidiol-treated mice, shown in red, have a far greater reduction in their average seizure frequency. So together, this suggests that cannabidiol is effective at reducing both the febrile seizures as well as spontaneous seizures in Dravet syndrome. So, is cannabidiol effective as a monotherapy? I believe that our data suggests that yes, indeed. In our mouse model that has both febrile and spontaneous seizures that are associated with Dravet syndrome and are consistent with what we see in human populations, yes, cannabidiol on its own, in the absence of other anti-epileptic drugs, reduces the severity and duration of febrile seizures and the frequency of these spontaneous seizures. So yes, it's effective as a monotherapy. But the next question is, how is it working? How is cannabidiol achieving this anti-epileptic effect? And so to address this neural mechanism, use techniques such as slice electrophysiology to gain this mechanistic insight. But before I jump in to the mechanism, I just want to point out that in a normal brain, you have a relative balance between excitation and inhibition, which is driven by, again, a relative balance between the signaling of the main inhibitory neurotransmitter GABA and the main excitatory neurotransmitter glutamate. This leads to seizure-free and normal behavior, but this is disrupted in Dravet syndrome. This heterozygous mutation affecting the SCN1A gene reduces the expression of NAV1.1 channels, which again is the dominant sodium channel expressed in inhibitory neurons, which I'm showing you here in red or pink. And this tips the brain's excitatory inhibitory balance towards excitation. Now, this was identified in our initial studies where we measured sodium currents in Dravet mice, finding that in pyramidal neurons, excitatory pyramidal neurons, there was no effect on the sodium current density in those neurons. However, there was a reduction in the sodium current density in inhibitory neurons. So you can see on the right, in representative current clamp recordings of inhibitory neurons where we're measuring the frequency of action potentials that are elicited with increasing amounts of current injection, Dravet mice are no longer able to sustain that high frequency of action potential firing that's needed to dampen the excitatory transmission in the brain at high levels of current injection. So there's this impairment in inhibitory interneuron activity, which tips the brain's excitatory-inhibitory balance towards excitation, leading to excessive excitatory transmission through the brain, causing seizures and a host of additional behavioral comorbidities. So therefore, I hypothesized that cannabidiol was having its anti-epileptic effects by selectively rescuing this excitability of these inhibitory interneurons. Now, I tested this effect using slice electrophysiology techniques where I was able to record changes in electrical signals in individual neurons. So I was able to record things like individual action potentials, which is a measure of cell excitability. And you can see on the left, representative trace of action potentials. Each one of these upward deflections is an individual action potential. And that's going to be consistent with neurotransmitter release. And I'm also able to record afferent neurotransmitter release from this presynaptic neuron as postsynaptic currents using voltage clamp techniques. And you can see on the bottom left, this representative trace of spontaneous inhibitory postsynaptic currents, which are these downward deflections that I've indicated with an asterisk. And these spontaneous inhibitory currents each represent the neurotransmitter release from the presynaptic neuron that in this case are detected by synaptic GABA-A receptors. Now, I began to test the hypothesis that cannabidiol was reducing excitatory transmission in the brain by recording from neurons in the dentate gyrus of the hippocampus. And I chose this region because the dentate gyrus is thought to be an important gate for excitatory transmission through the brain. And breakdown of this gate, so too much excitatory transmission from the dentate gyrus to the CA3 region of the hippocampus, is known to be associated with seizures in both humans as well as rodents. Now, you can think back to your introduction to psychology course many years ago. Remember the famous case of HM, who had bilateral hippocampal lesions that left him with the inability to form long-term memories. Now, the reason he had that surgery in the first place was because he was having excessive seizure activity, likely due to the breakdown of this gate. So therefore, I hypothesized that cannabidiol was having its anti-epileptic effects through reducing the excitatory output from the dentate gyrus. Now, I tested that hypothesis by making whole cell current clamp recordings of granule cells in acutely prepared slices from Dravet syndrome mice. Now, consistent with my hypothesis, I found that cannabidiol at a dose that's equivalent to the 100 milligram per kilogram dose that had anti-epileptic effects in vivo, reduced the frequency of spontaneous action potentials. So this is consistent with the idea that it's reducing excitatory output from the dentate gyrus to the CA3 region. Now, interestingly, this effective cannabidiol was blocked in the presence of the broad-spectrum GABA-A antagonist gabazine, therefore suggesting that this reduction in excitatory transmission is mediated by an increase in GABAergic transmission from these inhibitory interneurons. So to gain further insight into cannabidiol's effects on the interneurons themselves, I made whole cell voltage clamp recordings, again from granule cells and slices from Dravet syndrome brains, and measured the effect of cannabidiol on the frequency of spontaneous iPSCs in the presence of ionotropic glutamate receptor antagonists CNQX and AP5, just to isolate the effect of cannabidiol on on the interneurons themselves. Now, I found that consistent with cannabidiol increasing inhibitory interneuron activity, Cannabidiol increased the frequency of spontaneous iPSCs, which of course was blocked by gabazine, but interestingly was also blocked by the voltage-gated sodium channel antagonist, tetrodotoxin, suggesting that cannabidiol is increasing GABAergic transmission in an action potential dependent manner, which is consistent with it increasing the excitability of these inhibitory interneurons. So to test if cannabidiol is indeed rescuing the excitability of inhibitory interneurons, I made whole cell current lab recordings of interneurons themselves, specifically the parvalbumin positive interneurons, which are fast spiking brain cells that provide a great deal of inhibition to these excitatory granule cells. And you can see them labeled in the bottom right-hand corner of the screen. Now, I tested the effect of cannabidiol, shown here in red, on the frequency of evoked action potentials with increasing amounts of current injection. Now, I found that cannabidiol reduced both the rheobase, which is the minimum amount of current needed to elicit a single action potential and indicative of increased excitability, as well as increased the maximum action potential firing frequency at increasing current injection steps. So this fits with the previous data and suggests that indeed cannabidiol is rescuing this excitability deficit in interneurons of Dravet syndrome mice. But to gain insight into cannabidiol's effects on the granule cells themselves, I conducted a similar set of experiments where I looked at the effect of cannabidiol on action potential frequency in the granule cells themselves with increasing current injection levels. Now here, cannabidiol, again shown here in red, had no effect on the rheobase, unlike what it did in the inhibitory interneurons. However, it reduced the maximum action potential firing frequency at these high levels of current injection that the cell might see, for instance, during a seizure. So if we take all of this information together, it suggests that indeed cannabidiol is rescuing the excitability of these inhibitory interneurons, which increases GABAergic transmission to the granule cell and likely dampens down this excessive excitatory output from the dentate gyrus. This can be interpreted as perhaps reducing or preventing those spontaneous seizures from occurring. However, like in the case of these febrile seizures, when a seizure does occur and we see protection by cannabidiol, this could be acting by reducing the output potential of these granule cells themselves. So it's having dual actions both on the inhibitory interneurons and the granule cells. But I'm most interested in how cannabidiol is rescuing this excitability deficit in the inhibitory interneurons. So the question becomes, how is it achieving this effect? Now, we looked at a variety of neural targets, and many of you may know, but THC, for instance, is thought to act on CB1 receptors in the brain. And through action on CB1 receptors and activation of these receptors, it reduces calcium influx to the presynaptic neuron and thereby reduces neurotransmitter release. So one might hypothesize that perhaps cannabidiol was increasing GABAergic transmission by blocking or antagonizing these CB1 receptors. And indeed, it's been shown previously to antagonize the effect of THC on CB1 receptors. So if this was the mechanism of action in the brain, then a CB1 receptor antagonist should mimic the effect of cannabidiol to increase neurotransmitter release and occlude cannabidiol's effects from having further action on neurotransmitter release itself. So I tested this effect using the CB1 receptor antagonist, AM281, and found that yes, this antagonist mimics the effect of cannabidiol slightly by increasing spontaneous iPSC frequency in these granule cells. However, it was not able to block cannabidiol's further action to increase iPSC frequency. So this suggests that no, this is probably not the mechanism by which cannabidiol is having its anti-epileptic effects, because it still seems to be increasing GABAergic transmission through an additional mechanism. Now, another target, and there are many in the brain that have been proposed for cannabidiol to act on, but one that has been recently proposed as a potentially important target for cannabidiol's action is this G protein coupled receptor known as GPR55. And the endogenous ligand here is a lipid signaling protein, which I'll just abbreviate as LPI. And it's thought that cannabidiol can antagonize GPR55 and antagonize or block LPI's activation. So it's unclear, though, what effect this antagonism of the GPR55 receptor would have on inhibitory interneurons. But I hypothesize that if indeed cannabidiol was acting as a GPR55 receptor, perhaps this was the mechanism by which it was increasing GABAergic transmission. Now, again, if this is the case, then a GPR55 antagonist alone should both mimic the effect of cannabidiol to increase GABAergic transmission, and it should block or occlude cannabidiol's further action on any sort of the measures that we've been looking at so far. So I conducted a similar protocol of experiments, this time using the GPR55 receptor antagonist, CID, with a bunch of numbers, and I'll just abbreviate as CID here, but indeed consistent with the idea that CBD was acting as a GPR55 receptor antagonist. It mimics the effect of cannabidiol on its ability to reduce granule cell spontaneous action potentials. It mimics the effect of cannabidiol to increase spontaneous iPSCs, and mimics the effect of cannabidiol on these excitability measures in the inhibitory neurons themselves. And furthermore, it occludes and blocks the effect of cannabidiol from having any further action on any of these measures, together suggesting that this cannabidiol-induced rescue of inhibitory interneuron excitability is mediated by GPR55 receptor antagonism. So we believe that this is the mechanism through which cannabidiol is having its anti-epileptic effects, which we've shown to be working as a monotherapy to reduce both febrile and spontaneous seizures in our mouse model of Dravet syndrome. So how is it working? How is it acting as a monotherapy? Through antagonism of the GPR55 receptor and restoration of GABA-A receptor-mediated signaling. Now, the next question is, if it can protect against seizures, can it also rescue additional comorbidities associated with the disease? And I chose to focus my attention on autism in Dravet syndrome, in part because those with Dravet syndrome who are fortunate enough to make it into adulthood often see that the frequency, complexity of their seizures decreases over time. However, they're still left with a debilitating form of autism that severely impacts their quality of life, both for them as well as their caregivers. So any promising pharmacological treatment for Dravet syndrome should not only target the seizures in the disease, but also the autism-like social deficits. So to test the effect of cannabidiol on autism in our mice, I used the 3-chamber test of social interaction, where again, our mice choose to interact either with a novel stranger mouse or an object, in this case, an empty pencil cup. And like before, wild-type mice, which are shown here on the left in gray bars, prefer interacting with the stranger mouse compared to the object. Whereas Dravet mice treated with vehicle and shown here in this blue bar are practically indifferent, spending an equal amount of time interacting with the stranger mouse compared to the object. However, low doses of cannabidiol at 10 and 20 milligrams per kilogram rescued this sociability deficit. And these mice then spent a greater amount of time interacting with the stranger mouse compared to the object. This is exciting, but we also wanted to better understand cannabidol's effects on additional measures of social behavior, such as social reciprocity and how they respond to the social cues that are conveyed by other mice. So to do this, we use the reciprocal interaction test, in which two mice are placed in an open field chamber and allowed to freely explore the chamber and each other. Now we found, the mice choose to explore. They sniff each other. They make nose-to-nose sniff, nose-to-butt sniffs, nose-to-body sniffs. And the most exciting data that came from this experiment was that we found that our Dravet mice, especially those treated with vehicles, shown here in blue, engage in a great number of these defensive escape behaviors, which you can envision when a mouse goes to make a social interaction, and let's say it makes a nose-to-nose sniff. It quickly becomes overwhelmed within that social interaction and must quickly escape. And we see the mice quickly dart to the opposite corner of the chamber, where they huddle there and freeze, indicative of them being scared and anxious. So these Dravet mice seem to be highly anxious, and although they engage in a similar number of total social interactions, the quality of that social interaction is impaired. And again, they're engaging in a higher frequency of these defensive escape behaviors. But cannabidiol rescued that behavior, reducing their frequency of these defensive escape behaviors to around wild type levels. So this suggests that cannabidiol not only can rescue some of the sociability deficits, but can also recover some of these abnormal social behaviors that we're observing in our mice. But it may also indicate that it's having some sort of anxiolytic or causing a reduction in anxiety in our mice. So to test the effects of cannabidiol on anxiety, we use the elevated plus maze, in which, as the name implies, is simply just a maze that's raised off of the ground and illuminated from above. Now, highly anxious mice choose to spend a great amount of time in these closed arms, which are protected conceivably from threats in the environment, compared to the open arm. Now, as you'd expect, Dravet mice treated with our vehicle solution spend a great amount of time in the closed arm compared to the open arm. But a single acute dose of cannabidiol rescued this anxiety phenotype. Now these mice spend about an equivalent of time spent in both the closed arm as well as the open arm. So we find that cannabidiol not only rescues some of the sociability deficits that are associated with autism and Dravet syndrome, but also the anxiety-like phenotype. So they were able to now answer a number of these important scientific questions. We find that cannabidiol is effective as a monotherapy for treating seizures, as well as additional comorbidities in Dravet syndrome, including autism-like social impairments, as well as anxiety. So what might this mean for the treatment potential of cannabidiol in Dravet syndrome? Now, it's a very interesting and important question, because a lot of people fear that the seizures, this high frequency of seizures that occurs during this period when the brain is still developing, causes such irreparable brain damage that once it occurs, you can't really rescue through pharmacological intervention. But what our data suggest, at least in our mice, is that a number of these large behavioral comorbidities that are associated with Dravet syndrome are merely the result of this imbalance between excitation and inhibition in the brain. And if you can restore that balance through pharmacological intervention, in this case cannabidiol, which is increasing GABAergic transmission, well, then you can rescue a number of these behavioral abnormalities. So this is potentially very exciting for both patients as well as their caregivers, who through cannabidiol might see beneficial effects not only on reducing seizures, but also on some of the additional comorbidities that come with the disease. So to summarize what I've demonstrated so far, we find that cannabidiol on its own is effective at reducing the severity of the febrile seizures in Dravet syndrome that characterize the early stage of the disease. And we also see that it reduces the frequency of those spontaneous seizures that characterize later stages of the disease. We find that this likely occurs through a restoration in the excitability of these inhibitory neurons, which occurs through CBD antagonism of GPR55 receptor activity in the dentate gyrus of the hippocampus. This increases GABAergic transmission within this important brain region, dampening the excitatory output from the dentate gyrus and preventing those spontaneous seizures from occurring. We also find that cannabidiol in adulthood could be a promising therapeutic technique to also rescue some of these behavioral comorbidities associated with Dravet syndrome, including autism-like social impairments and social anxiety. So despite these, what I believe are exciting results, I think the research is far from complete. And there are a number of future directions that I'm pursuing currently and looking to pursue in the future as I begin in laboratory up at Western Washington University at the end of the summer. And they really stem from this important question, which I think is the major elephant in the room that many of you are probably thinking, okay, we have anti-epileptic effects of cannabidiol at high doses, which I've shown you that it's effective at 100 and 200 milligrams per kilogram, and ineffective at low doses at protecting against seizures. However, it's achieving these benefits in rescuing social behaviors at much lower doses, about 10 or 20% of the effective dose for treating epilepsy. So how do we go about dosing? And it does present a problem. Now, cannabidiol, because of the range of targets that it acts on in the brain, has what seem to be like these specific therapeutic windows for different types of disorders. You might call them the Goldilocks zone, whereas cannabidiol is only effective at treating epilepsy with certain doses of cannabidiol that are different than its benefits in treating autism and anxiety, which are only effective or effectively treated with cannabidiol at other doses. And cannabidiol's benefits in treating those disorders is lost when you leave that Goldilocks zone, if you will, when you leave that therapeutic window. So this creates a problem, and we're looking at ways of overcoming that. Now, one of the ways is through targeting the GABAergic signaling mechanism through essentially different or multiple mechanisms. So I've told you about cannabidiol's effects to increase spontaneous GABA release, which is mediated by synaptic GABA-A receptors and is responsible for this traditional phasic inhibition characterized by the fast rise time and quick decay, which I've shown you so far. But there's also a powerful form of tonic inhibition, which is mediated by extrasynaptic GABA-A receptors that contain the delta subunit. And that's represented in this highly filtered trace on the bottom. Now, these extrasynaptic GABA-A receptors are thought to be non-desensitizing and therefore contribute a large degree of the overall inhibitory charge to the cell. And so therefore, it represents a powerful therapeutic target to increase inhibition in disorders that are otherwise deficient. So through potentially combinatorial therapeutic approaches involving both cannabidiol and perhaps modulating levels of endogenous cannabinoids like 2-AG, we can target both phasic inhibition as well as tonic inhibition to greatly increase that level of inhibitory function in the brain and perhaps rescue both seizures and autism-like social deficits using the same dose of drug. So just some preliminary data that we're excited about. We've been testing the effects of increasing 2-AG levels by blocking the hydrolyzing enzyme ABHD6 using a novel inhibitory drug, KT182, and we find that this drug increases the magnitude of the tonic current in Dravet mice to even almost a greater extent than that seen in wild-type mice, suggesting that these mice, as well as presumably Dravet syndrome patients, have a greater tonic current targeting possibility for pharmacological intervention. So perhaps a combinatorial approach using both CBD as well as drugs that increase 2-AG levels might be a promising therapeutic strategy for achieving behavioral rescue of both seizures as well as other comorbidities simultaneously in Dravet syndrome. So with that, I'd like to thank a number of people I've worked with on this project. Dr. Bill Catterall, who sponsored the initial stages of this project. Dr. Ruth Westerbrook in the lab has been a really important colleague for bouncing scientific ideas off of. She's also the one who conducted the seizure studies, looking at the effects of cannabidiol on both febrile and spontaneous seizures. I'd like to also thank my current mentor, Dr. Nephi Stella, who I'm working in his lab now to continue on these studies. And together, all of them for helping me get to this point where I'll be beginning a lab, as I mentioned, at Western Washington University to carry on this, what I believe, important and exciting research in cannabidiol, as well as the larger cannabis field as a whole. With that, thank you for your attention. And if there are questions, I believe that I'll be getting those through email, and I look forward to answering those. All right, thanks for your attention.
Thank you, Dr. Kaplan, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, why do you feel the need to treat autism?
Yeah, that is a really good question. And look, there are many different forms of autism. Autism is a spectrum disorder after all, and people can fall on all across that spectrum. Individuals with Dravet syndrome tend to have a very severe form of autism, to the point that they really struggle to function in daily life. This is not the individual who just likes to paint a lot or read a lot of books or do math. This is a very debilitating form that not only can be harmful for the individual, but also is concerning for the parents. Parents often report that one of the things they're greatly concerned about with their children is they're unable to pick up on these critical social cues that keep them out of harm's way, and as a result struggle and can get into accidents and other harmful things. So we're talking about a very severe form of autism. Often that might have impacts on causing self-injurious behavior and other things. So we're not talking about trying to cure all forms of autism, but help those individuals who struggle in such a way that it severely detracts from their quality of life.
Thank you. The next one is, did you test the effect of CBD directly on excitatory transmission in the brain?
Yeah, so for timing's sake, I didn't show you all the data that we have collected, but we also looked at the effect of cannabidiol on excitatory postsynaptic currents and other types of excitatory measures. Now, we find that cannabidiol selectively increases the excitability of inhibitory interneurons, not the excitatory neurons themselves. And some of those data I already showed you, but if we look at the effects of cannabidiol, for instance, on spontaneous excitatory postsynaptic currents, we do see that cannabidiol reduces these EPSCs. So it's reducing excitatory transmission. But interestingly, this effect is blocked in the presence of GABA receptor antagonists. So this reduction in excitatory transmission seems to be occurring through an enhancement in inhibitory neuron signaling. So yeah, good question.
Great, thank you. How did you measure seizure severity? Is it possible that the CBD treated mice were having seizures that you didn't detect?
Yeah, that's a great question. Yeah, so we did this all by eye. So we used a Racine scale, which is out of 5, 5 being the most severe. Now, it's really hard to detect seizures at level 1 or 2. They're not these overt physical symptoms. You would have to use an EEG. And we have done EEG studies in the lab before, just never with cannabidiol. So we look at seizure severity in our mice by eye just at a seizure level of 3, 4, or 5. And we see that on average, cannabidiol is reducing the severity of those seizures from a Racine level 5 to a Racine level 3, 3.5. But as you note, it is possible that they're still having seizures at a Racine scale of 1 or 2 that we're just not detecting. And future studies are, we're planning future studies where we're going to implant EEG electrodes to see if cannabidiol is also reducing some of those lower, overtly undetectable seizures. Yeah, great question.
Thank you. Okay, we have time for one more question, and it is GPR55 antagonism also the mechanism for treating autism?
Well, again, a good question. So we looked at the effect of essentially cannabidiol at the concentration that was having anti-epileptic effects, so about 16 micromolar. This is higher than the effect that we're seeing, and we know that 16 micromolar dose is essentially acting through GPR55 antagonism to increase inhibitory neurotransmission and presumably reduce seizures. We did not look at this lower dose of cannabidiol, so we don't know if this is the same mechanism, for instance, that is contributing to the pro-social effects that we see and anti-anxiety effects that we see. We are currently engaging in studies in the lab where we are injecting this GPR55 antagonist drug, the CID drug, ICV into the brain. It doesn't seem to cross the blood-brain barrier, so we're injecting it directly and looking at its effects on social behavior and some other behaviors. So don't have an answer for you directly on that question, but it is certainly one we are interested in pursuing. Now, I would just like to note that there are many other therapeutic targets by which cannabidiol is having its action in the brain, including serotonin receptors, as well as TRPV1 receptors and CB1 receptors themselves. And so other groups have found that a lot of the anxiolytic benefits of cannabidiol in the brain are through increasing serotonergic signaling, through acting on these 5-HT receptors. So it wouldn't necessarily be surprising if there is a role for GPR55 in the pro-social anti-anxiety effects, that it's not the only effect, because presumably it's also having these beneficial behavioral effects through actions, again, on serotonin receptors and other targets.
That is great. I would like to once again thank Dr. Kaplan for his presentation, but also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 29th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.